Hello to all. Today we are going to discuss some MCQs which are based on the cell biology. This is the part 3 and already I have uploaded two parts, part number 1 and part number 2 on my channel. So just go through that questions also. These questions may be very important for the forthcoming NEET exams. And if you are new to the channel, so please subscribe the channel and also like and share the videos. So starting today with the very first question, and that is the diagrammatic representation of chromosomes is called as. Now this is very important, and sometimes I have seen that the students commit mistake between these two options, ideogram and karyotype. Always they are having the confusion between the ideogram and the karyotype. Holotype and homotype are totally different. They are not at all associated with the chromosomes. But the diagrammatic representation of the chromosome is known as, always remember that if we are representing the chromosomes diagrammatically, then it is ideogram. But if we are arranging the chromosome according to their morphology, that is size, shape and all, then it is called as what? The karyotype. If we are arranging the chromosome according to the size, shape, alright, then it is karyotype. But the diagrammatic representation, or in fact I will say the diagrammatic representation of the karyotype, the diagrammatic representation of the karyotype is basically what? The ideogram. Is it clear? So the diagrammatic representation of the chromosome is always called as the ideogram. So the question number first is having the answer A. Now question number 2, in set chromosome. You know what is set chromosome? A set chromosome is a chromosome which bear satellite at its terminal part. Right? All chromosomes are not having the satellite. All chromosomes are not having the satellite. So in set chromosome, set is terminal part of the chromosome. In set chromosome, set is the terminal part of the chromosome. Alright, say for if this is the chromosome last part, then this is the set part, alright, last part. So the set or the satellite is terminal part of the chromosome beyond secondary constriction. Now this is beyond, this is secondary constriction. So what happens that beyond secondary constriction, a part of the chromosome is pinched off out, and this part beyond the secondary constriction, which is pinched off out, is actually called as what? The satellite or the set. So the question is that in set chromosome, the satellite is terminal part of the chromosome, that we know very well, beyond secondary constriction. Now the question is this, that this satellite consists of. So always remember that this satellite consists of. Question is DNA, options are DNA, RNA, repetitive DNA, or none of these. So the satellite, which is the terminal part of the chromosome present beyond the secondary constriction, consists of the repetitive DNA, and these repetitive DNA will play a very important role when you will be studying the DNA fingerprinting. In DNA fingerprinting, this is having a very important role. Repetitive DNAs or the satellite DNAs form the basis of the DNA fingerprinting, alright, because these repetitive DNA sequences have polymorphism in them, alright. So the satellite, which is the terminal part of the chromosome, consists of the repetitive DNA, alright. And these repetitive DNA or the satellite DNAs are also of different types, microsatellite, alright, mini satellite, etc. So question number 2 is having the answer C. Question number 2 is having the answer C. Now coming to the question number 3, which among the following can be seen only under electron microscope? Now when we use electron microscope, to view those things which cannot be seen by our naked eye, So definitely that thing must be very much small. That small thing will only be seen by the electron microscope. So which among the following can be seen only under the electron microscope? Chloroplast. This is very much big. We know very well that chloroplast is the the organelle which is having the size 4 micron to 8 micron. A ribosome is having the size of 150 angstrom to 250 angstrom only. Leucoplast are again a type of plastid and they are big in size, and nucleus you know very well, it is very big in size. Okay, so now the question rises that which among the following can be seen only under electron microscope? So ribosome is the answer, because ribosome are the smallest membraneless cell organelles. Remember this thing, ribosomes are the smallest membraneless cell organelles. They are the smallest membraneless cell organelles, are having the size of 150 angstrom to 250 angstrom only, and that's why they are the smallest cell organelle and they can be seen only under electron microscope. Question number 3 will be having the answer B. Question number 4. In eukaryotic cell, the question is only for the eukaryotic cell. The type of ribosome is, now many times, though it is very simple question, but due to haziness, some people will commit a wrong answer for this question, right? Only 70S? No, 70S ribosomes are found in prokaryotic cell. Only 80S, 70S as well as 80S, or only 50S? Now what the students do, that they opt 80S, but the third option is having 70S and 80S option both. Answer must be 70S as well as 80S. Why? Because generally the eukaryotic cell contains 80S ribosome, no doubt at all. But this is also truth. That the eukaryotic cell also consists of mitochondria and the chloroplast. Eukaryotic cell also may also consist of the mitochondria and the chloroplast, and both the mitochondria and the chloroplast are the feature of the eukaryotic cell. And in mitochondria and the plastid, that is the chloroplast, 70S type of ribosomes are found. 70S type of ribosomes are found. So definitely our answer will be not only 80S, the answer will be 70S and 80S, because generally in eukaryotic cell 80S type of ribosomes are found, but eukaryotic cell also consists of chloroplast and mitochondria, and they have 70S type of ribosomes also. So question number 4 will be having the answer C. Next, in cell division, whenever we study the cell division, we see that spindle fibers are formed. And what is the prime role of the spindle fibers? The prime role of the spindle fibers is to carry the chromosome towards the opposite poles. Now the question is that spindle fibers are made up of which protein? So always remember, a spindle fiber is made up of 97% part. Remember this thing, 97% part. This is very important information, that 97% part of the spindle fiber is made up of tubulin protein, and only the remaining 3% part of the spindle fiber is made up of RNA. Alright, so a spindle fiber is made up of 97% tubulin, which is a protein, and only 3% part is made up of the RNA. So question was for the protein only, that spindle fibers are made up of which protein? So definitely it is straightforward, it is written in the B option that it is made up of tubulin protein. So question number 5 will be having the answer B. Alright. Now question number 6, bulk of histone proteins synthesized in. Now you know very well that histone proteins are basic proteins. Alright, if you might have heard about H1, H2A, H2B, H3 and H4. Alright, H1, H2A, H2B, H3 and H4. There are 5 types of the histone proteins, and during the cell division you have studied the cell cycle. If you are not studied, you can go through my video in which I have explained the entire cell division. Alright, so bulk of histone protein. Now you know very well that in cell cycle there is interphase stage, and that interphase stage is completed in 3 phases, G1, S and G2. Just after G1, that comes the S phase, and in S phase 2 important processes occur. One is the DNA replication and another is the synthesis of the histone proteins. Always remember, that in S phase of cell cycle, what happens? Two important processes occur. One is the DNA replication, that's why it is called as the synthesis phase, and also there is synthesis of the histone proteins. So the question is, bulk of histone protein synthesized in G1 phase, G2 phase, S phase or G0 phase. So always remember it is synthesized in S phase. So question number 6 is having the answer C, and that is known as the S phase. Alright. Now question number 7, synapses occur during. Now have you ever heard about this word synapses? Now there is a big difference between synapses and synapse. Don't be confused by these two terms. Alright, synapse word was used in the nervous system, right? Synapse word was used in nervous system, alright, where the junction where the two neurons meet, that junction was called as the synapse. But we are not talking just now about the synapse, we are talking about the synapses. And the literal meaning of the term, synapses is pairing, So the question is, synapses occur in dash phase of meiosis. We must know very well that there is a phase known as meiosis first. There is a stage known as meiosis first, very important stage. And in that meiosis first, there is a stage known as the prophase first, which is the longest phase of the meiosis first. And in prophase first, there are 5 substages, if you are remembering. LZPDD, again I am repeating, LZPDD, leptotene, zygotene, pectine, diplotin and dikinesis. Again I am repeating, leptotene, zygotene, pectine, diplotin and dikinesis. And it is a sequential process. Are you getting? It is a sequential process. So what happens that in leptotene, attractive forces are increased. But in zygotene, more attractive forces are more between the chromosome, and because of the attraction between the chromosomes, the two homologous chromosomes come near to each other. The two homologous chromosomes come near to each other and they show pairing. So this pairing of homologous chromosome, remember this thing, that this pairing of homologous chromosome in the zygotene substage of prophase first of meiosis first is actually the synapses. So synapses is pairing of homologous chromosome, and it always occur in zygotene stage. So the answer of question number 7 is zygotene. Synapses occur in zygotene phase of meiosis. Alright, so question number 7 is having the answer A. Question number 8, term basal body is associated with the development of. Now, if you have heard about cilia and flagella, so always remember this thing, that the origin of the cilia and the flagella always happens from the basal body. Say for, if I am drawing here a diagram, say for I have drawn the flagellas. So these flagellas are originating from the basal body. These flagellas are originating from the basal body. So the term basal body is associated with the development of cilia and flagella, cell plate, phragmoplast and kinetochore. So the best option is term basal body is associated with the development of the cilia and the flagella. So question number 8 is having the answer A. Is it clear? So today we have discussed MCQs on cell biology. This was part 3. Alright, and for your preparation, to give you extra edge for your NEET preparation, I have uploaded lots of videos based on MCQs on my channel, as well as I have also uploaded many videos based on the theoretical concepts. Okay, so you can go and watch that. So thanks a lot for watching me.